So I did just want to introduce SmartStax really briefly to give the basic introduction and set the context for where we are and why we're here this evening. Because of course our three panel members will talk in detail about this issue and this technology. And I wanted to let you know that it was in July that Monsanto and Dow AgroSciences announced that they had received approval, regulatory approval, in the US and Canada for this new corn, this new genetically modified corn called SmartStax. And it is a unique corn or a unique GMO in that it's stacked to contain 8 GM traits together. And there are stacked traits with 2 or 3 traits on the market. There have never before been 8 traits stacked together. And this is what we see here. And this is produced through crossbreeding of GM plants. And our panelists will discuss what that entails, what some of the risk questions might be. And aside from other issues of patent control, where here you have 8 patents in one seed, market control, where Monsanto, of course, is the largest seed company in the world. And there are some very real issues for farmers' choice as early as next year when SmartStax will be grown. And for pricing, that there will be a price increase of over 40%, according to Monsanto, for them bringing this technology together in one seed. It was the Canadian Biotechnology Action Network that investigated the regulation behind SmartStax. Seeing that this new product was approved, we then asked the question of, well, what was the decision-making process to approve this product? And we saw that, in fact, there's really no documentation available from either Health Canada or the Canadian Food Inspection Agency to detail that decision. Typically, there are decision documents, which are summaries of those decisions, that the Canadian Food Inspection Agency, the CFIA, and Health Canada publish. But the totality of the information available to us on SmartStax was 3 paragraphs on the CFIA website. We did write to both the CFIA and Health Canada and the Minister of Health with our various concerns. And we're happy to say that the CFIA did take the time to meet with us at some length to discuss the issues and has agreed to publish the list of stacked trait products, these GM stacked trait products, that have been approved thus far. Because if you were to go to the CFIA and Health Canada website, you would find lists of plants with novel traits and novel foods, which is the category within which the Canadian government regulates genetically engineered crops and foods. But you would not find any of the stacked products listed. And this comes to the heart of the issue, or at least the basic of the issue, which is that the Canadian government has not assessed SmartStax as a stacked trait product because Health Canada and CFIA have already approved the 8 traits, so-called novel traits, individually in previous crops and foods. And so the issue here is that our regulatory system does not see any risk issues with the 8 being combined together. And I did just want to flag that, well, the terms are confusing, but we will use the term genetic engineering and genetic modification interchangeably, GMOs or GE products. In the regulatory terms, really, we would be talking about genetic engineering. So just to summarize then, the CBAN, the Canadian Biotechnology Action Network, has requested that the Minister of Health withdraw approval for SmartStax. Really what this would mean is requesting the CFIA to withdraw approval, because actually there's no paperwork that we can see that Health Canada did in relation to SmartStax. So we are continuing to ask for some response from the Minister of Health, and we continue to dialogue with the CFIA, which is much appreciated. So it's my great privilege tonight to introduce the panel. Dr. Steinbrecher is a biologist and geneticist. She is an invited member of the Federation of German Scientists and the British Society for Allergy, Nutritional and Environmental Medicine. She has acted in capacity as a scientific expert in many governmental and public consultations and even court cases. And she's been closely associated with the UN-led international negotiations in the implementation of the Cartagena Protocol on Biosafety. And Dr. Steinbrecher has been doing that work since 1995. And actually, Dr. Steinbrecher right now is part of the ad hoc technical expert group of the biosafety protocol that's looking at risk assessment of GMOs, and particularly the subgroup on stacked trait products. Dr. Steinbrecher specialized in gene regulation since 1982 and has worked as a research scientist in the field of mutational analysis, gene identification, and gene therapy in university and hospital settings. She's the author of many scientific papers, reports, briefings, commentaries, and at present is co-director of a non-profit public research organization in the United Kingdom called EcoNexus. Please welcome Dr. Steinbrecher.
Stacking. Stacking of genes. Here, stacked GM events are defined as those events which combine GM events in form of conventional breeding. For example, if you have conventional corn and you genetically engineer them or transform them, as we say, with gene constructs, the constructs, for example, could be A, B, C, and D, and then you would have, if they are transferred individually in different plant lines, you would have like 4 different events resulting from it. A corn with the gene cassette or the traits of A, then B, C, and D. From that, you would now do some crossings. For example, you could cross A and B, and then you have the plant AB as a result, and C and D, then you would have the DC as the result. And in the next generation, or whenever you're ready, you would then breed those two in order to get to the next generation that combines all 4 events. Of course, it's much more complex in the breeding than this looks, but I'm not going into that area. I just wanted to have it roughly sort of pictured so you see what we're talking about. SmartStax is based on 4 parental transgenic lines, which is like a Monsanto 89034 line. I will not read all the numbers, so you can see we have different lines here which have been combined through breeding, and this line at the bottom is actually the unique identifier for SmartStax if you go to the biosafety protocol site or the European Union sites in order to. In this case, we have a combination of 2 herbicide tolerance genes, which is the usual glyphosate resistance for Roundup and the glufosinate resistance or tolerance for Liberty pesticides. I think that is the brand names which I used here. Then we have 6 further genes involved, which are all derived from a bacterium called Bacillus thuringiensis, which are endotoxins. Now, how do we go about with the assessment? What's been done so far? The idea is that if you assess them individually, once they have been genetically modified, once you have the event, and you look at, is the gene present? Is it fully there? Is it only 1 copy? Do a few other tests. So that is assessing it individually. So you can do that for A, for B, for C, for D. And then you can start with your process. And according to the assessments done here or also in the US, this is then no further tests required as the original events have been assessed. Now, this is a very interesting case because if, as a regulator, one then, or also as a scientist, says like, this is sufficient, that's where it ends, one actually has to really trust some very clear assumptions. If this approach, as I said here, if this approach and methodology is chosen, it relies deeply on numerous assumptions to be correct. If they are not correct, then we have a problem with this approach. So that's what I will be looking at the assumptions and if they are correct.